Hello and welcome to our single-cell multiome analysis series with SciDAP. In the previous video, we were reviewing our dimensionality reduction analysis for ATAC-Seq, and this time we are starting the cluster analysis. So we are creating a new sample and choosing the pipeline that's called single-cell ATAC-Seq cluster analysis. We give it a name. I name it Step 6 ATAC Clustering. Then we select one of our previous samples. In this chain of dimensionality reduction and cluster analysis for both RNA and ATAC-Seq, we could consequently pick the previous step, so the analysis goes smoothly, taking into account the data that was generated before. So we select our Step 5 dimensionality reduction. Then we select our aggregated sample from the beginning. For target dimensionality, again, you can pick anything. Let's put it to maximum. And just as in the case with RNA-Seq, we could do multiple resolutions. Then you could select Find peak markers. This function is important if you will be annotating your clusters with using your ATAC-Seq and genes of interest together. It may take additional time to process, but it is very helpful. They will be highlighted on the UMap. We will see it in the next video. Then let's select a few marker genes. Then, in Advanced tab, you could choose Show HTML Report, pick a color scheme, and you can leave this as a default. Then just click Save and Run. And this is it. In the next video, we will review the output of this single-cell ATAC clustering. Thanks for watching.